and welcome. Today we are discussing this question from GATE examination 2023, paper Life Sciences, question number 98. The question states, the presence of excess glucose has been known to prevent the induction of lac operon as well as other operon controlling enzymes involved in carbohydrate metabolism in E. coli. Which of the following processes defines the phenomenon? A. Catabolite repression, B. Attenuation, C. Glucose effect, and D. Feedback inhibition. It will be good to remind you that this is a MSQ question, so there can be multiple answers correct. To discuss the answer of this question, we also must discuss the lactose operon or the lac operon. So, lactose operon, it is discovered in the prokaryotic cells in E. coli, for example. So, first of all, let us first talk about operon. What is an operon? So, an operon is a group of genes that are situated consecutively under the control of the same promoter. So there is one promoter which controls the transcription of some consecutive genes, right? So they are all under the control of the single promoter, and the mRNA transcript of these genes are all contiguous. So that means that there is a single mRNA that is produced for all of these genes. From that single mRNA, different proteins are produced. Now, this is an example of the gene regulation in the prokaryotes. This is an example of gene regulation in the prokaryotes. And here we are discussing about the lactose operon or lac operon. So what does it do? As the name suggests, that the lac operon is meant for metabolizing lactose, the milk sugar, right? So we know that E. coli, this bacteria, it resides in our gut, right? And so it's a healthy gut microbe, and whenever we drink milk or any milk products, we consume this milk sugar, which is lactose. So therefore this lactose can also be utilized by our gut bacteria. Now the point is that we know that bacteria usually readily utilizes glucose for metabolism because glucose is a monosaccharide. Glucose is a monosaccharide, whereas lactose is a disaccharide. So if the bacteria has to utilize lactose or has to metabolize lactose, then it has to first break down the disaccharide into two monosaccharides, right? And then it can metabolize lactose. So for breaking down lactose, the bacteria needs energy. So therefore, it is important for the bacteria to spend energy before breaking down lactose to obtain energy from it. So it is not a very readily available source of energy for bacteria. So if glucose is present in our gut or in the media, the bacteria will always first try to metabolize glucose. And in absence of glucose, if lactose is present in the gut or in the medium, then bacteria will try to utilize lactose as well. Now, therefore, the bacteria will need genes for metabolizing these lactose. So these genes will basically synthesize some enzymes which will be essential for the lactose metabolic pathway. All right. Now, these genes are these three genes, the LAC-Z, the LAC-Y, and the LAC-A. Now, these three genes produce three different products. I should write it down here that LAC-Z produces the enzyme beta galactosidase. LAC-Y produces beta galactosidase permease, and LAC-A produces, here I should write beta galactoside permease, and LAC-A produces beta galactoside transacetylase. So these are the three enzymes that these genes encode. Now, I am coming to the function of these genes, but we have to remember that these genes are under the control of a single promoter region. Now, there is also another cis regulatory site in this gene construct, and that is called the operator site. So this operator site is basically downstream to the promoter. So it's on the 3' side of the promoter, and then there is also a cap site. I will come back to this later. So what is the necessity of the lactose operon? To synthesize these enzymes for metabolism of lactose. Now, as I mentioned, that bacteria will not like to metabolize lactose when glucose is present. Why? Because glucose is simply readily available energy, and it does not have to break down glucose into any simpler compound or any simpler component. So therefore, when glucose is present, bacteria will not likely metabolize lactose. Right? So therefore, the lactose operon has to be functional only when lactose is present, or only when bacteria can access lactose. So therefore, it is essential that this lactose operon has a switch on, switch off system. So when is it switched on? It is switched on, the lactose operon is switched on when lactose is present and glucose is absent. Whereas, so I'll write it here, lactose operon is on when lactose is present and glucose is absent, whereas the lactose operon will be switched off when lactose is absent and glucose is present, or when glucose and lactose both are present. So which means if glucose is present, whether or not lactose is present or absent, the lac operon will be shut off. Because bacteria has glucose, it does not need any lactose metabolizing enzymes. So therefore, there has to be a mechanism where the gene transcription is switched on and switched off. So that is maintained by two regulatory components of the lac operon. These two regulatory components, there are actually two regulatory components, two types of regulations. One is a positive regulation, another is a negative regulation. So I will first discuss about the negative regulation. So this negative regulation is brought about by a protein which is called the repressor protein. The repressor protein. What it does? It shuts off the transcription when glucose is present, right? So, or when, rather to say more correctly, repressor shuts off the gene transcription when lactose is absent. Alright, when lactose is absent. So this repressor protein is actually coded by a gene which is called LAC-I. Coded by the gene LAC-I. So LAC-I is also part of this operon system, but it is actually situated far away from the promoter region. So there is a gene LAC-I that produces this repressor protein. We know the LAC repressor. Okay, and what is the function of this repressor? The repressor actually binds to this operator region. So it is shown here, the repressor binds to this operator region. And how does it help? So the RNA polymerase, we know it binds to the promoter region of the genes, right? And from the promoter region, it starts transcription and it goes in the 3' direction, right? So now if the operator site is blocked by a repressor, so if the repressor is sitting at the operator site, it is bound at the operator site, then the RNA polymerase will not be able to move forward. So it is facing a constraint here. It is not being able to move forward, and if it is not being able to move forward, then of course there will be no transcription from these genes. Now the question is, when does the repressor bind to the operator? So the repressor is actually synthesized continuously. Okay, it is a continuous expression of the repressor. So the repressor is produced all the time, and the repressor will bind to the operator all the time, and it will continuously shut off the transcription from the LAC genes unless and until there is lactose. So what lactose does is that when this lactose can actually bind to the repressor. Lactose or any other analog of lactose, like for example allolactose here shown, it can bind to the repressor, and because of that the repressor changes its conformation, and then the repressor cannot bind the operator site anymore. So the repressor will not bind to the operator anymore. So here the operator's region becomes free, and the RNA polymerase can now move ahead, can now move forward, and now can synthesize the LAC genes, right? Can now transcribe the LAC genes. So therefore, now there will be transcription from these 3 genes, the LAC-Z, LAC-Y, and the LAC-A gene, right? So basically, what is turning the LAC operon on? The lactose or any other analog of lactose, right? So that is why it's called a negative regulation. So usually the lactose operon is shut off, but switched off, but when there is lactose, it is turned on. How is it turned on? By the repressor protein. So the repressor protein moves away from the operator region, and the RNA polymerase can now start transcribing the LAC genes. So that is the concept of the repressor protein. Now we will also look at this cap site. What is the role of this cap site then? And that is part of the positive regulation of the LAC operon. Now this is called catabolite repression or glucose effect. So as I mentioned that whether or not glucose whether or not lactose is absent or lactose is present, if glucose is present in the system, in our gut, in the medium, if glucose is present, then the lactose operon will always be shut off, right? So the glucose has an effect on this regulation, on this lactose operon, right? As that is only justified because glucose is a better source of energy than lactose, right? So if glucose is present, there is no need to metabolize lactose. So how is this what is the molecular mechanism of glucose being present and how it shuts off the lactose operon? So actually, when glucose levels are low, there is a production of cyclic AMP. So CAMP is cyclic AMP. Now cyclic AMP has a binding site on the DNA, and that is the cap site. Cap is basically catabolite activator protein binding site. So that is the cap site, catabolite activator protein binding site. So I'm sorry, the cyclic AMP does not bind to the cap site, but the cyclic AMP binds to this protein which is called cap. Cap is the cap is the catabolite activator protein, right? Cap is the catabolite activator protein. So cyclic AMP binds to the catabolite activator protein, and then this cap-cam complex, the cap-cam complex binds to the cap site on the DNA. Now actually, cap is a positive regulator of the RNA polymerase. So it helps in the RNA polymerase to bind tightly to the promoter, and then it results in high levels of transcription. Okay, so basically cap-cam complex is a positive regulator of the RNA polymerase. So when cap-cam is bound, it results in high levels of transcription from the lactose operon. And when is it happening? When the glucose levels are low, because only when the glucose levels are low, cyclic AMP is produced. Okay, so when glucose levels are low and there is high transcription from the lactose operon, which is justified. And what happens when the glucose levels are high? When glucose levels are high, no cyclic AMP is produced. So if there is no cyclic AMP, then cap cannot bind to the cap site without the cyclic AMP, and therefore transcription occurs only at a very low level, right? So basically, when there is glucose present, there is low transcription from the lac operon. So which justifies our concept that when there is high glucose, the bacteria does not need to metabolize lactose, and therefore it does not switch on the lactose operon. So that is how the other catabolite, which is glucose, represses the lac operon. So actually this catabolite repression stands for the concept that when glucose is present, which is a readily available source of energy for bacteria, it represses the catabolism of other components, right? So that is why it's called a catabolite repression or glucose effect, and that is the positive regulation for the lac operon. So in the negative regulation, the repressor protein is ensuring that only when lactose is present, the genes will be transcribed, and the glucose level and as well as the cyclic AMP levels are ensuring that only when glucose is low, then the transcription will occur, right? So that means that whenever there is lactose but there is also glucose, the lactose is present, so the repressor does not bind. So here in this particular scenario, the repressor is bound to lactose, and therefore the repressor does not bind to the operator, right? The repressor does not bind to the operator, so the operator region is free, so the RNA polymerase can move ahead, but there is no cap-cam complex. So even if the RNA polymerase can move ahead, but it will result in a very low level of transcription. So therefore, practically, when there is glucose, the lac operon is not switched on. Alright, so now coming back to our question. So which of the following processes define the phenomena? So it is actually the catabolite repression. So option A is correct. Attenuation is not something that we had discussed here, but attenuation is part of the tryptophan operon. If you study the tryptophan operon, you will study it. And C, glucose effect. That is also correct. So it is basically the same. It is these two terms, the catabolite repression and glucose effect, they are referring to the same event, the same phenomena. And the last one is feedback inhibition. That is also not correct. So our answers are A and C. All right. So that's it, and thank you.